Thank you very much. That was awesome introduction. Okay. Can everybody see my screen now? Are we good? Kim, I can see you clearly, so if you can stay on, I can use as my litmus. Needless to say, Kim, thank you so much for that. Very nice. Here, I'm going to do one thing real quick. Let's see. Stop video. I really appreciate that very nice introduction. It was great. And I just cannot tell everybody how wonderful it is to be back here. It's just been fantastic. And I look forward to working with all of you again. So moving straight ahead with this seminar, I'm going to talk to you about some work. The first set of data was just published or coming out in Glia, and then another set of data was published a while back. We have some new data on this. Uh-oh. How do I advance my slide? Oh, hang on. Okay, great. So as many as you know, as Kim indicated, most of the work that we're doing in our lab really is focusing on looking at molecular and cellular mechanisms that drive neuroinflammation, neurodegeneration, and really within the past 5 years, repair in response to microbial infection of the CNS. But we've really been devoted a lot of our work to mouse coronavirus infection. And so we really have three main areas of research going on in the lab right now. We've been working a lot on microglia. This has really stemmed from some of the really great work out of Kim's lab and Matt Blurton-Jones. In addition, we became really intrigued in how neutrophils may impact neuropathology in response to viral infection of the CNS. And then finally, we've done a lot of work with how surgical engraftment of neural progenitor cells, either mouse or humans, can influence the immunological microenvironment that then can promote repair. But for today's talk, I'm really going to talk primarily about microglia and our neutrophil story. So this is funny because I'm sure you know a lot more now than you did before about coronaviruses. We have been working for over 20 years on a neuroadapted strain, which is the JHM strain of mouse hepatitis virus. JHM simply stands for John Howard Mueller. He, back in 1949, isolated the virus, or they weren't really isolating viruses at that time, but he had a paralytic mouse in his colony and had the wherewithal to take the brain and spinal cord out and homogenize it and pass it through a filter. Coronaviruses, as you no doubt know, are positive sense single-stranded RNA viruses. The cartoon depiction is shown here. So our strain of virus is in the beta coronaviridae family, which is actually the same as SARS-CoV, MERS-CoV, and SARS-CoV-2. You have in this cartoon depiction extending furthest from the surface, this is the spike glycoprotein, which mediates attachment to cell receptors. So in humans, it is ACE2 for SARS-CoV-2, as well as the other SARS I mentioned, or SARS and MERS. There's an interesting paper that just came out in bioarchives that suggested another protein, CD147, is another receptor. So I think we're waiting for independent verification of that. In addition, you have other surface proteins, the hemagglutinin esterase, and then membrane glycoproteins, and then the nucleocapsid is associated with the viral RNA. So our strain of virus, if we do an intracranial inoculation of susceptible mice, and in this case, it's primarily black 6 animals, these mice will develop an acute encephalomyelitis. The virus that we're employing is glial-tropic, so it has very little to no ability to replicate in neurons, at least not very efficiently. The animals that survive the acute phase of the disease will develop an immune-mediated demyelinating disease, both in the brain as well as in the spinal cord. And they get, this correlates then with mild to severe hind limb paralysis. And so, you know, I put this slide in here because I think it is relevant for the time that we're living in. And so coronavirus is really, your context and the majority of the world's context is thinking about them as upper respiratory tract infections. And so OC43 and 229E they can account for between anywhere from 15 to 30% of upper respiratory tract infections during seasonal periods of time. However, there's clear evidence that these viruses, along with MERS-CoV and SARS-CoV-2, can infect and replicate within the CNS. And I get some of the early work that was done on this was done by a person named Pierre Talbot, who was a postdoc in Mike Buckmeyer's lab. And he's had a very good review that came out in early December before this whole thing hit about human coronaviruses and other respiratory pathogens and their ability to gain access into the CNS and cause disease. Now, relevant to SARS-CoV-2, there have been almost on a daily basis an increasing number of reports of patients that have been affected by either exhibiting neurologic symptoms, including headache, dizziness, loss of taste and smell, meningitis, as well as encephalitis, and then in one extreme case, a necrotizing hemorrhagic encephalopathy. And also stroke has also been reported in some individuals. So it really argues for that there is a neurologic component that is associated with SARS-CoV-2 infection. So with that in mind, for the past 20 years, we've been employing this model of mouse coronavirus to infection of the CNS to understand mechanisms that drive host defense as well as disease. But I would argue that these data or findings might be relevant to SARS-CoV-2 induced neurologic disease. And so that's kind of where we're pivoting. But I can show you the data we have now on the mouse coronavirus system, and I'm anxious to see how it works in the human. So very quickly, we do an intracranial inoculation with our virus. It's not the most natural route of infection, but it does a great job. And if you look here in panel A, you see viral titers peak at around 5 to 7 days post-infection, depending upon the dose of virus. You see a very early and rapid mobilization of components of the innate immune system, characterized by neutrophils and NK cells and macrophages, but it's really virus-specific T cells that are sensitized in the draining cervical lymph nodes that then expand there and then migrate into the central nervous system following a chemokine gradient. Neutralizing antibody doesn't really come up until much later. But this is important because the neutralizing antibody is critical in preventing viral recrudescence within the CNS. Now, with regards to the demyelinating disease that you see, again, the viral titers come up and then go down below the level of detection. And in both circumstances, why I say that, that's by plaque assays. But you can detect viral antigen in the CNS by immunohistochemical staining or viral RNA by in situ hybridizations, sometimes as far out to about a year post-infection. And it's this chronic phase that the animals develop this immune-mediated demyelinating disease. And this is just an old in situ hybridization that I did when I was in Mike's laboratory. And this is H&E staining showing a white matter track, and these black silver granules are the viral RNA. And then sequential sectioning of this section stained with Luxol fast blue, you can see numerous inflammatory cells and the ensuing demyelination that occurs. So it's where the virus is persisting that results in a chronic elaboration of pro-inflammatory cytokines and chemokines. This brings in virus-sensitized T cells as well as macrophages and potentially microglia that contribute to white matter damage. This just shows how rapidly the virus can spread given the route of infection. Again, this was done back in 1998, right before I joined the faculty here when I was in Mike's lab. But this is in situ hybridization. This is the brain of a mouse. So you can see the olfactory bulb here and then the distinct anatomic structures. This is 3 days post-infection. And you can see very early, again, this is the intracranial inoculation. The virus is replicating in ependymal cells, and then it gets into CSF, and it gets in discrete locarias in the olfactory bulb, as well as in the hind area of the brain. But by day 7 post-infection, you see this massive expansion of virus throughout the brain. Now, correspondingly, this is just showing that we see a very rapid response by the innate immune system, characterized in this case by a chemokine that I have a lot of passion for, which is CXCL10. This is expressed in almost identical areas in which we see the virus. Again, by the ependymal cells lining the lateral ventricle, we see it in the olfactory bulb, and then here extending into the brainstem. And then again, we see this massive expansion of CXCL10 at day 7 post-infection. And the cell types that are primarily responsible are astrocytes. And so this is just co-staining for GFAP and CXCL10 mRNA transcripts. So in response to the infection of the central nervous system, you see quite a bit of pro-inflammatory gene expression. Chemokines are elaborated very quickly. And this is actually very true if you look at the literature on SARS-CoV as well as SARS-CoV-2. CXCL10 is one of the first chemokines that comes up in the lungs and then is detected very readily in the blood. And then finally, we see this robust demyelination. These are spinal cords taken from control animals prior to infection. And then this is day 28 post-infection, stained with toluidine blue. You see the loss of the blue staining in the lateral white matter columns here. And you can compare that to the control animal. These areas that we box out are used for electron microscopy. It's a very powerful but laborious technique to look at demyelination. You can see in the control animals, these are axons that are coming out towards you. This is a lot of, quite a bit of myelin that's circulating. This is what you would expect in an uninfected animal. Here are axons that are stripped of the myelin sheath. And the reason we do a lot of EM is mainly because if you want to look at remyelination, you have to do EM analysis because that's the only way you can calculate what's known as a G ratio to look at new wraps of myelin. And then finally, several years ago, right before we left for Utah, we were working with Milton Greenberg and Mike Holland's lab and employing 2-photon microscopy to look at various aspects of spinal cord pathology and repair. And one of the things that really jumped out at us in that paper was the fact that we could see axonal damage. This is just Thy1 YFP expressing axons as early as 7 days post-infection with the virus. So here's axons in a non-infected animal, and then one axon over time. And so we start seeing this degeneration. It really has led to some interesting ideas that we're looking at. Early axonal damage may actually precede demyelination. So demyelination is immune mediated. T cells and macrophages amplify demyelination. We do not see epitope spreading, so that is the generation of myelin reactive T cells in this particular model. At least they might be there, but they're not driving the neuropathology of disease. So this kind of summed up a lot of work that we've done, and of course a number of other people have done in the field. And it certainly highlights that control of viral replication within the central nervous system is very complicated and involves this very regulated orchestration of the innate and adaptive immune response. And then along these lines, neuropathology, that is axonal damage and demyelination in the face of this persistent infection of the CNS, is mediated by glial responses that work with components of the immune system. And so we've employed a lot of techniques to study this, including a lot that I've shown you: flow cytometric analysis, in situ hybridizations, immunostaining, 2-photon microscopy. But within the past year, we've started employing single-cell RNA sequencing on sorted CD45 positive cells isolated from the brains of these coronavirus-infected animals to understand really what might be happening at a more granular level. And so very quickly, you all understand how this works. This was done at the Huntsman Cancer Center at the University of Utah, and they do have a very good genomics core there, I must say. But the idea is very straightforward. We are taking animals at 0 days post-infection, or controls days 3, 7, and 21. We remove the brain and spinal cord. The brains were done at 0, 3, and 7. The spinal cords were done at 21 days. The reason we selected these time points in brief, because we needed the sufficient number of cells at day 0 to do the analysis. Day 3 represents the great time for the innate immune response, as I showed in that graph. Day 7, this is when virus-specific T cells have infiltrated into the CNS. And then day 21 is where we have spinal cord demyelination. So that's the underlying reason we selected these time points. We do a standard Procol gradient, discontinuous Procol gradient centrifugation to isolate lymphocytes and CD45 positive cells. We flow sorted these cells out, did the barcoding, and then we get our classic Tisney plot to do the analysis. This just shows you here again kind of what I showed you. These are the animals. This is work done by Amber Sage. For some of the data, I'll just show you. For this experiment, the animals all got sick, which we expected them to. Viral titers were in the brain. Spinal cords had demyelination. And then down here are the cell numbers that we isolated at each time point. So from the uninfected brain, day 3, day 7, and day 21. So again, this is pooling all of these cells. And then this is the number of reads per cell. And this is actually very important because this is the depth of the analysis, right? So doing this, this is actually aggregate data of all time points, 0, 3, 7, and 21. And This isn't a Tisney plot. It's kind of this next generation is called a UMAP, which I really like because you can actually bin the cells out, and it's easier to kind of just sort your way through it. So for example, the dendritic cells are shown here, T lymphocytes are shown here, microglia here, monocytes, macrophages, and B cells. And then also we get some neutrophils coming in. But what really jumps out is from the work that we did with flow cytometry, we certainly see these populations of cells, but it's the heterogeneity that's really impressive. So for example, these combinations of dendritic cells that we observed at defined times post-infection, we did not know that there are this diverse population. Similarly, these different macrophage and monocyte subsets. It was very intriguing. And then finally, I'll touch on this because this is going to springboard us into the rest of the talk. This just shows we had to do the bioinformatics approach. And again, Amber Syed in the lab has been tremendous in doing this analysis. It's a Seurat analysis with an R package. Essentially, this is what is called a dot chart. And you probably are all very familiar with this. But simply the size of the dot represents the frequency of cells within the cluster. And then the color intensity basically indicates the level of gene expression down here. So in this capacity, we can just, again, bin out these different cell populations based on their gene expression profiles. And it's been very, to date, pretty accurate. And then I'm going to just show you two other slides before getting really into the data. But the reason I'm showing you this is because it validates the integrity of the single cell RNA sequencing approach to understand what's going on at a kind of a real molecular level. And it's consistent with what we observed with flow staining. And I just bring your attention here. So these colors just indicate different time points. So turquoise is uninfected, green day 3, red day 7, and pink day 21. But you see this natural increase in inflammatory CD4 T cells, effector CD8 T cells, these cycling CD8 T cells. What's intriguing is work by Connie Bergman and others have found that antibody secreting cells are enriched primarily at later stages post-infection. We see that. And then, and then finally, this ebb and flow of dendritic cells that comes up and then down, which I think is very interesting. So I feel confident about these data, and we're kind of looking forward to moving forward on this. And then finally, again, we can use these Tisney plots to bin out our cells and then really start interrogating different gene expression profiles at defined times post-infection. So it's a very powerful approach, as you all know. Oh, and then lastly, what I also like is you can look at the ebb and flow of interferon responses. So because this is a virus, very early at day 3 post-infection, we can see in microglia, macrophages, and dendritic cells an enriched type 1 interferon response, or interferon alpha. And again, work by Connie Bergman and others have really emphasized the importance of these cytokines in controlling coronavirus infection or replication within the CNS early. And then if you recall, by day 7, we have antigen-sensitized T cells infiltrating into the CNS, and they're secreting copious amounts of interferon gamma. And so now we see these interferon gamma responses in these cells. So the one thing that we also became very interested in, this is actually what Amber was noticing when she kept doing this, is these different subsets of microglia that appear at defined times post-infection with virus. Now, these could be the same microglia and they're just having different gene expression profiles. But to us, this was very interesting and implied that microglia might have a very important role in either host defense or disease. And so just a real quick sidestep on microglia, this audience probably doesn't need it that much, but these are quote unquote the tissue resident immune cell of the central nervous system. For years, they've been considered important in driving a number of different human and CNS diseases, including Alzheimer's, MS, traumatic brain injury or spinal cord injury, as well as psychiatric conditions. And there's been a lot of traction within the field using different drugs to target microglia because of the increased penetrance of certain drugs that can selectively get to microglia and get rid of them. And so the idea is that in normal white matter tracts or in normal gray matter tracts, you have just quiescent microglia that kind of do their job on a daily basis. But in response to pathology, so for example, here in an AD brain, you see a dramatic increase in genes that are expressing factors that might be contributing to disease. The same is true in models of multiple sclerosis, which is, of course, an inflammatory autoimmune demyelinating disease. We also see microglia expressing factors. There's actually a lot of crosstalk of these disease-associated molecules in the gray matter as well as the white matter. And so I think that's also very interesting. So then, as I said, we became very interested in what microglia might be doing within the context of a neurotropic viral infection. And so we wanted to address this by depleting microglia using a drug called PLX 5622 that targets colony stimulating factor 1 receptor. And this is required to maintain microglia integrity and maintenance within the central nervous system. And the model is quite nice, and you're probably familiar with this from all the work that Kim's done, because he really kind of pioneered this work. But we can take experimental animals prior to infection and feed them either control chow or Plex 5622 containing chow. We do this for 7 days, and then we challenge the virus intracerebrally with virus. We took brains out at day 7 post-infection, sorted out CD45 positive cells, and did the single cell RNA-seq on this. And then we removed the spinal cords at 14 days post-infection and did the exact same thing. Real quick, this is just some representative flow data showing that prior to feeding animals the Plexicon 5622, microglia were present both in the brain and in the spinal cord. But at the start of the experiment, we had dropped the levels of this dramatically. And importantly, we do not see any loss of myelin integrity following Plexicon-treated uninfected animals. So here's what happens when we feed the animals the Plexicon 5622. In the hashed lines, we see this dramatic drop in survival compared to control animals. And looking at 3, 7, 12, and 14 days post-infection within the brain, at 3 and 7 days post-infection, we see a significant increase in viral burden, which is maintained, yet not at significant levels, out at 12 and 14 days. By day 21 in the surviving mice, we couldn't detect virus in either the brain or the spinal cord. And then finally, this is just flow analysis of the brain at day 7, and then at day 14 in the spinal cord, showing that we've depleted microglia successfully in these animals using different stains, so CD45 or CD11B, which is a well-accepted marker. All right, so what's happening with T cells? Because this is a viral infection of the central nervous system, and T cells are critical in controlling viral replication. And again, it's through either interferon gamma secretion in response to JHM infection or cytolytic activity. And here is just a representative flow from day 7 in the brain, gating on CD4 T cells and CD8s. This is in the control animal or the Plexicon-treated animals. And what's interesting, and if you just enumerate these, we see a gradual increase in the number of overall CD4 T cells and a significant increase in the total number of CD8 T cells. And then if we do tetramer staining to look at virus-specific CD4 or CD8 T cells, again, we see a slight increase in the virus-specific CD4 T cells as well as virus-specific CD8 T cells. And we're kind of scratching our head about that because, again, at day 7 and at later time points, there are increased numbers of these T cells, yet there's an increased number of virus or amount of virus in the brains. And I'll get back to that in a minute. And this is why, again, the single cell RNA-seq was very helpful. So here is a Tisney plot at day 7 comparing the infiltrating cells in the control animals as well, and that's in blue, as well as in Plexicon-treated animals. And one intriguing aspect, this is again Tisney showing that we did see successful depletion of microglia, consistent with our flow analysis using well-accepted markers for our transcripts associated with microglia, including TM, EM119, P2RY12, and SPARC. And the thing that really just I was surprised about was that it didn't really alter this immunological landscape in terms of at least infiltrating immune cells and or microglia, except that we got rid of microglia. The one standout here is these B cells, right? Because we see this larger population of B cells in the Plex-treated mice versus the control animals, which we need to look into in more detail. We haven't done it yet. It's just one of those things that once we get up and running again here, we're going to start revisiting that. but because the T cells are so important in controlling replication of the virus, we started looking at transcripts associated with activation states. And so the t-SNE plot shows here our CD4 T cells. Again, this is at day 7. We see really no difference in expression of CD69, but we do see reduction in transcript levels for other activation surface markers such as CD44, interleukin-2 alpha and beta receptors. And then this is very interesting. This is TBX21, which encodes for TBET. So this was dramatically reduced to transcript levels in these T cells. Now, in contrast, if you look at the CD8 T cells, and again, here they are in the t-SNE plot, we see increased levels of granzyme B, PD1, as well as perforin. So you have these differential activation states. And I can get into some of the reasons why I think this is what's happening, but one of the things that really jumped out at us was if you look at macrophages, which work by Stan Perlman and Connie Bergman and earlier Steve Stolman, clearly showed that macrophage antigen presentation was very important in controlling or helping and control replication. And we see these decreased levels of transcripts for MHC class 2 in macrophages, as well as CD86 in the brains of plex-treated animals versus control animals. Yet in dendritic cells, there was really no difference whatsoever, and in fact, slightly higher levels. And we confirm these observations by looking at day 7 by flow cytometry, and then we're starting to look at this in more detail by immunohistochemical staining. So in other words, at day 7, we see this muted expression of potential antigen presentation by a subset of immune cells, specifically the macrophages, that may impair the ability of the T cells to efficiently control virus. And if we look at MHC class 1, though, what's interesting, again, in the macrophages, it's about the same, if not slightly increased. And on dendritic cells, there is no difference whatsoever between Plexicon-treated animals versus control animals, okay? So it seems to be made primarily in Plexicon-treated mice. The MHC class 2 expression is altered in macrophages, but no other APC that we saw. And then MHC class 1 expression is really not affected at all either. So finally, one of the real reasons I wanted to do these experiments was to look at the neuropathology, specifically demyelination, that could occur. And because I would have thought that microglia would have been very important in contributing to demyelination. But what was surprising is we saw an increase in the severity of demyelination upon Plexicon treatment of these animals. So in other words, when we got rid of the microglia, we see much more severe spinal cord demyelination, and this is quantified here. And then we did EM analysis and found that, also surprisingly, that remyelination was impacted in these animals. And so this is a control animal with an infected animal that was not treated with Plexicon. And these are just thin myelin sheaths that are wrapping around these axons. But in contrast, in the spinal cords of the Plexicon-treated animals, we saw numerous axons that were very swollen and with very little to no remyelination. And again, we're going back and calculating what's called this G ratio, which again, what you do to calculate a G ratio is you measure the diameter of the axon and divide it by the diameter of the axon plus the myelin sheath. So the higher you are to 1, the less remyelination you have. And so remyelination was limited according to our G ratio analysis, and then the overall thickness of the myelination or the myelin was reduced as well. So this suggested that microglia were potentially restricting the severity of demyelination as well as augmenting or enhancing remyelination. And so to look at this in more detail, again, we went back to our data set for the single cell RNA-seq, and not to walk through this in much detail, but what we found is a number of genes that have been associated with these disease-associated factors in numerous neuroinflammatory disease models, including Alzheimer's and EAE, which is the prototypic MS model. And in these, what we found is in different subsets of macrophages, there's an increase in, for example, ApoE, which encodes for apolipoprotein E, this transmembrane glycoprotein NMB, which is actually very, very interesting literature on that and demyelination. Osteopontin, there's increased transcripts for this. Larry Steinman at Stanford is a big advocate for this molecule and potentially driving disease. And then TREM2 is increased as well. So in other words, when we get rid of microglia, the inflammatory macrophages exhibit increased expression of transcripts that are associated with demyelination. And then if we look at well-defined molecules associated with remyelination, we again see subsets of macrophages that we see a dramatic decrease in expression of, for example, cystatin F, insulin growth factor 1, and then lipoprotein lipase. And there's a very compelling body of literature that out in the field that suggests that these molecules are critical for allowing remyelination to occur. And so we're actively pursuing this because I think there's some very important and compelling questions that would impact how we view remyelination and the role of microglia in this process, or these complicated processes. So for this portion of the talk, I would argue that microglia aid in control of viral replication within the CNS, in part through antigen presentation or influencing antigen presentation. And this aids in the stimulation of infiltrating virus-specific T cells. When we remove or get rid of microglia, we see an increase in demyelination, and that's associated with this unique macrophage subsets or subpopulations that have distinct mRNA signatures that might be contributing to disease progression or increased demyelination. And then correspondingly, remyelination efficiency is negatively impacted when we get rid of microglia, suggesting that a population or subpopulation of microglia may be important in augmenting repair by regulating expression of molecules associated with remyelination within macrophages. So there's a lot of work for us to do on this, but this is the best place for us to do this type of work. So I'm excited to move forward on this.
Tom, I have a question. This is Craig.
Oh, hi, Craig.
Hey, did you guys look at oligodendrocytes or OPCs to see if there were differences after PLX treatment?
No. Well, yeah, we started doing it by staining. And so we do see a lower number of oligodendrocyte progenitor cells. But the thing that we need to do is nuke-seq and to really get a better handle on that. We've done staining, Craig, but we've also done employed our nanostring. The nanostring had a company or has a new kit, a neuropathology kit. So we've employed that, and it is interesting. We see it based if their signature profiles are correct, and Ken might be able to elaborate on this in more detail. But we see a dramatic decrease in transcripts associated with oligodendrocytes and OPCs, arguing that we might see an effect. I don't think the plex is affecting the oligodendrocytes directly. I think it's probably an indirect effect, if anything.
Yep. Thank you.
Okay, I just have a couple more slides and then I'll be finished. To be honest, and no offense, Eric, I never really considered neutrophils that, it's a viral infection, right? So, but they are intriguing. And then we started looking at some of the papers in the clinical literature in a little more detail. And the reason why we started doing this is because people with progressive forms of multiple sclerosis, their disease-modifying therapies that are FDA approved have no effect on, or very little to no effect on disease progression in these patients, arguing that most of these therapies are targeting the adaptive immune response, for lack of a better description. They're trying to really limit access of potentially antigen-sensitized lymphocytes, both T cells as well as B cells, from gaining access into the CNS and forming new lesions. But the fact that people with progressive forms of the disease don't respond to these drugs argue that perhaps components of the innate immune response might be driving neuropathology. And so there's some interesting articles that have come out showing that neutrophils are associated with clinical disease progression as well as demyelination. And certainly preclinical mouse models of MS, including EAE, and then toxin models also argue that neutrophils can increase neuropathology. And so, but it's really not been looked at in our viral model of demyelination. So we became very interested in this. And so to accomplish this, our question was to sustain neutrophil infiltration into the CNS, augment demyelination in JHMV infected mice. I want to emphasize sustain because normally neutrophils come up very early following infection and then they go away. But they are very important because we published a paper in PLOS Pathogens a while back showing that if you block neutrophils from getting in early, what happens is you don't have the fenestrations in the blood-brain barrier, and this then limits access of antigen-sensitized lymphocytes. But we wanted to keep the level of neutrophil infiltration up and to see what happened. And so we generated a transgenic mouse in which allowed us to do this. These are the animals right here. I won't belabor this point, but we could cross, we made these animals when I was back here at UCI before going to Utah. But essentially what it is, we've generated mice that in adult animals, we can inject doxycycline, and this generates increased expression of CXCL1 transcripts in astrocytes. So first, CXCL1 is a chemokine that's a very potent chemoattractant for neutrophils. The reason we selected CXCL1 is because we've done a lot of work on chemokine expression in the brains of JHM infected mice, and this was a predominant chemokine expressed very early. And then finally, we knew that astrocytes, from our studies as well as others, that CXCL1 is derived primarily in astrocytes during periods of neuroinflammation. So our model was very simple. We just wanted to verify that, that these animals, that they worked. I'm kind of condensing some data, but essentially we took our transgenic mice, and so I just wanted to go back. The nomenclature we used is abhorrent, and so let me just point this out. Double transgenic mice are those animals that respond to doxycycline and overexpress CXCL1. So they have the GFAP responsive promoter, whereas the single transgenic only have the CXCL1 transgene, but you can't overexpress it, okay? All right, so here we have our mice. We infect them with virus, and then we treat with doxycycline daily for 10 days. And then what we did is we took brains and spinal cords out from our experimental animals. So we were treating single transgenic with dox as well, by the way. And what we found is that in the brains, as early as 4, 7, and 12 days post-infection, we can see an increased level of transcripts for CXCL1. The same is true within the spinal cord. If you go in and stain for CXCL1 and GFAP, we saw what we wanted, which was a dramatic increase in co-localization of CXCL1 in astrocytes that from the double transgenic mice. And then finally, if you look at protein levels in the spinal cord, there's an increased level of CXCL1 in the spinal cord compared to the dox-treated single transgenics. And then also we saw quite a bit of CXCL1 in the plasma, which is not surprising because, again, the virus doesn't go out into the periphery, but remember, chemokines are soluble chemoattractant cytokines, essentially, and so they will be expressed within the CNS, and that will get out into the periphery. Now, here's what was really surprising with these animals. When we infected them, we saw this dramatic increase in mortality in the double transgenic mice treated with dox compared to the single transgenic mice treated with doxycycline. And anytime you see that in a viral model of disease, that's usually associated with impaired ability to control viral replication. But if you look here, what we found is that there really was no difference whatsoever. Sorry that I think this label's off, but these are the single transgenic with dox, these are the doubles. We don't see any change really at all in viral titers in the brain or in the spinal cord. And for the sake of time, we did not see any effect of dox-induced CXCL1 on either total CD4 or CD8 T cell infiltration, infiltration of virus-specific CD8 T cells, or macrophages or circulating monocytes. And I'll get back to why I think that is, and I'm interested if anybody else might have an idea on this because it could be an interesting project. But anyway, so this is just contour graph showing lymphocyte, or excuse me, neutrophil infiltration into either the brain or the spinal cord at day 12. And what was interesting is the dox treatment of either single or double transgenic, we didn't see a dramatic increase in newts in the brain. It did increase, And so this is showing the overall frequency here, as well as total numbers. But the spinal cord, it was a dramatic increase in the number of neutrophils infiltrating in the spinal cords. Again, here's the frequency as well as total numbers. And I think that might have to do with simply that the CXCL1 concentration level is enriched in the spinal cords compared to in the brain. And we can go back and actually test that. But also what was interesting is normally when we were looking at the neutrophils getting into the CNS following JHM infection, mainly the cells were meningeal. They didn't really penetrate in the parenchyma, and very few neutrophils were in the spinal cord. But here, at 12 days post-infection, what we can see is lots of CXCL1 staining in the double transgenic mice treated with doxycycline, and that correlates with nice penetrance of neutrophils into the parenchyma. And so this is just using a marker Ly6B2. And we actually could enumerate that by counting at different anatomic regions of the spinal cord. So either the cervical, thoracic, or lumbar. And you see these enriched areas or these anatomic regions in which in the double transgenic mice treated with dox, increased number of neutrophils infiltrating in. And then correspondingly, we saw a big increase in demyelination in these animals. And that's quantified here. Representative Luxol fast blue staining is shown here. And so this is the double transgenic animal treated with dox. You might not be able to see it so well, but these areas outlined in red, hash marks show areas of demyelination, which are extensive in the ventral tract, but also extending out into the lateral tracts, whereas at 12 days post-infection in the control animals is primarily limited right here in the ventral tract. And then what we did is, again, similar to looking at where the neutrophils were infiltrating in, we looked at these different anatomic regions, so T3, T5, 7, 9, et cetera. And where we saw the increase in neutrophils infiltrating into the spinal cord, there was a significant increase in the severity of demyelination. And then when there are areas where there wasn't that much difference in neutrophils or very low levels, we didn't see a big difference in demyelination. And then somewhat to Craig's point earlier about oligodendrocytes, using a marker for mature myelin-producing oligodendrocytes, which is GST-Pi, we saw a reduction in the number of these cells by immunohistochemical staining, and then correspondingly, an increase in the number of microglia. So this is just EBA1 staining. And then we saw this increase in microglia by flow staining. So there's some change here that's occurring. And then finally, we wanted to make sure if neutrophils were either directly or indirectly responsible for this white matter damage in the spinal cord. So we ablated neutrophils using an anti-Ly6G antibody. This just shows that this works very nicely because in the double transgenic treated with dox, we did not see any neutrophils infiltrating in compared to the isotype control. And then we also saw this reduction in the severity of demyelination in these animals, arguing that neutrophils can augment or at least have some impact on white matter damage. So this is in the JHM model. We also did this in EAE. So again, this is experimental autoimmune encephalomyelitis. You probably all know about this. And we saw almost the exact same results, that CXCL1 overexpression in these double transgenics increased white matter damage following induction of EAE. And this was published in the European Journal of Immunology. All right, last 3 slides. We're really trying to define mechanisms now by which this could be happening. So we're going back to the employing single cell RNA sequencing to see if we can see unique gene signatures in neutrophils that are present in the CNS of dox-treated double transgenic compared to controls. This just again is a UMAP showing it's really an amalgamation of the data that we have at day 7, or no, I think it's at day 12. Sorry, just simply day 12. And again, there's a spinal cord, the number of mice, the number of cells and reads per cell. And this is work that is being spearheaded by Dominic Skinner, who's in the lab right now. And just again, to show you, we're just getting these data. Like we moved here, we were hitting the data, and then, we're getting the lab set up, and then COVID-19 happened. And so we're kind of gradually resurrecting ourselves. But this is just the UMAPs from single transgenic mice compared to doubles. Again, it verifies our flow data in the spinal cord. We see this dramatic increase in neutrophils compared to what we see in single transgenics. We also see some modulation in other cell populations, so this might give us insight in what might be happening at a molecular level. We characterized our neutrophils. These are just violin plots. So neutrophils express a lot of CXCR2, which, by the way, is the receptor for CXCL1, which is another reason why we chose that chemokine. And then these are S100, A8, and A9 are calcium binding proteins that have some role in regulating inflammation, and neutrophils are a very prominent source for these molecules. So right now we're buying drugs to try to block these molecules to see if they have any effect on disease. And then the last data slide, what was interesting, and this is Dominic kind of just followed his lead on this. He looked at monocytes and found that infiltrating monocytes in these mice, the spinal cords expressed high levels of CXCL10 and CXCL9, and then these cells had an enhanced interferon gamma. Oh, excuse me. So again, these are the chemokines that attract T cells, and we've shown an important role for both these molecules in driving neuropathology. And then these either CD8 T cells shown here or CD4 T cells shown here, we see this increase in interferon gamma response in these two populations of CD8 T cells, as well as interferon gamma responses in CD4 T cells. And I think this should be interferon gamma, but I need to check with Dominic. So this also gives us a potential avenue to pursue in looking at how neutrophils may get into the CNS. There's certainly a neutrophil monocyte dialogue that can occur. There's literature on this. They enhance antigen presentation, but it might augment CXCL10 or CXCL9, and this could increase T cell infiltration into the spinal cords that also could be driving neuropathology. All right, so overexpression of astrocyte-derived CXCL1 resulted in an increase in mortality in JHM-infected mice, but it's not due to any impaired control of viral replication. The CXCL1 in the CNS increased neutrophil mobilization from the bone marrow. I didn't show you this data, but we see a dramatic increase in the blood. And this results in increased neutrophil infiltration in the CNS, and it correlates with enhanced demyelination within the spinal cord. Targeting neutrophils attenuated the severity of white matter damage, and as I alluded to, we're now really trying to find out what mechanism or mechanisms are occurring by which neutrophils can increase the severity of white matter pathology. So these are the people that did the work. Some of you may remember Brett. He started out my lab here when I was at UCI, came to Utah. He did a postdoc with, I think he was Mike Oldstone's last postdoc. And now he's at J&J in San Diego. Let's see, Dominic Skinner. Oh, Brett made the CXCL1 double transgenic mice. Dominic Skinner has taken these animals and is going to do some really great work with that. Vrishali Mangali was a PhD student of mine at Utah. She's now doing her postdoc at IU. She did a lot of the microglia studies. Amber Sage did all of the RNA-seq studies in conjunction with Vrishali's work. Yuting Cheng is also doing some really nice work with microglia that I wasn't able to touch on too much today. And then finally, Colleen Stone started in Eileen Anderson's lab and Brian Cummings' lab as a tech. She came out to Utah with me, and now she and her husband moved to UC Davis and are doing great there. And then finally, I just want to acknowledge funding sources that we have, some of the people at both UCI and Utah, and then some of the great colleagues I've had here that I'm looking forward to continuing working with. And then Ryan O'Connell and June Round, Adekon Ikez, and Bob Fujinami they were wonderful colleagues I had at University of Utah. And we had a lot of fun working together and publishing some nice papers. So I will stop there now. And then I don't even know. Oh.
You can leave it up.
Okay.
As you wish, because certainly we can go back to a couple of these things. So I sent around a note to just send questions by chat, and Mike says, in fact, Mike, why don't you just ask it, and Craig as well.
Okay, I'm unmuted, I guess.
Yeah, I can hear you.
Okay, Tom, have you determined or tried to determine the specificity of those infiltrating B cells? So the reason I ask is because in chronic LCM, CNS disease, you get essentially monoclonal B cell populations in the brain that actually form little colonies in the brain.
Yep. That is a great question, Mike. And luckily, when we first started doing the single cell, we were doing 3' sequencing. But for that data set, we did 5' sequencing. And so I believe we can start looking at the innate. I know we can do it for T cells. And I think we can do the same thing for B cell receptors. And so we can start looking and see if that is a clonal population.
Greg?
Yeah, so Tom, my question has to do with the observation of this increased population of neutrophils in these CXCL1 double transgenic mice.
Yeah.
You guys see, I mean, so I think your hypothesis is that has something to do with just increased T cell infiltration, but another possibility is maybe that those neutrophils are getting activated there and, you know, causing some damage of their own. So have you guys looked at neutrophil nets to see if there might be evidence for that in those spinal cords near the lesions?
Yeah, and we did that ad nauseum. There was a very good group at Utah that had really worked out detailed staining protocols for this because it's not trivial. And then secondly, they identified a unique drug in amniotic fluid, believe it or not, and that was selectively ablated net release in neutrophils. And we could never get really robust staining for nets in the spinal cords or brains of these double transgenic animals. And if we gave them that drug, it didn't really have an effect on the neuropathology. So I don't think it.
So Tom,
I wanted it to be that net mechanism, but I'm not
following
sure.
Following up on exactly that, though, can you go back to the single cell RNA-seq profile, the UMAP?
Yeah.
Profiles of the that show the neutrophils in there.
Do I do new share or what do I do?
Back.
Okay. Oh, I know.
No, just go back to
yeah. Okay, hang on.
to the slide.
Yeah. Okay.
You know, one of the things I thought about that as well when you were showing this, but I also yeah, right. I mean, but no, right there is good.
Right here.
Well, there or the next one. Yeah, this one. I mean, you know, you're not looking at a whole lot of cells here. This is when you're saying 200 neutrophils. This is of what? I mean, this is
that is out of the
whole brain slice.
Yeah.
Or
Yeah.
How many cells? How many CD45 cells? Positive cells.
It's on the CD45 sorted cells in the spinal cords out of. So
I mean, there's relatively few there. So I mean, first of all, I wouldn't expect to see nets with so few neutrophils there. I'm actually surprised that you're seeing this phenotype of the demyelination that's associated with neutrophils because.
Yeah, but I want to, you know, to their point, I got to check and make sure. I want to talk. If Dominic's watching, maybe he could elaborate on this and maybe clarify because there has to be more than 200 cells because by flow we see a ton of these cells.
Oh, okay.
So I need to make sure I did the UMAP.
Doesn't look like much as far as the
Right.
maps are concerned. The other thing I would be interested in seeing, you know, Milton's wife, Elise, was a postdoc in my lab until a couple of months back. But she was doing single cell RNA sequencing on neutrophils following infection. And she found all kinds of interesting subgroups of neutrophils. So I'd be interested to see, you know, what you and Dominic are finding in terms of what these neutrophils themselves are expressing besides the S100A9 is, that's a calprotectin. That's mostly a zinc, blocks zinc uptake and chelates zinc. But you know, it's made by the ton by neutrophils. So I'm not surprised you're seeing so much of that there. But I'd be curious to know what else.
What was the infection you were using?
Sorry?
What was the infection model you're using for your neutrophil studies?
Either fungal, aspergillus, or bacterial pseudomonas.
Okay.
Obviously, we see a lot more neutrophils. They take up much more. And this is corneal infections.
Yeah.
But yeah, we saw all kinds of interesting subgroups of neutrophils. So with this, you know, every gene that's being expressed, and it might be fun to.
I'd like to see those data. Okay, we can talk later about that. All right.
This is Tali. Can I ask a question?
Oh, hi, Tally.
So wonderful. Thank you. As a single, probably non-immunologist in a group, I was wondering if you've looked at all the gene expression properties of the astrocytes, which seem to be the main targets of the virus, or any other non-inflammatory cell group. Can they be altering the signal to the inflammatory cells, or can they themselves lead to demyelination through simply changes that are directly induced by the virus, rather than everything going through the beautiful network of innate and adaptive immunity that you're talking about?
Yeah, they're a major player in the demyelination. They're a prominent expressor of CXCL10, this T cell chemoattractant. And we've shown that we could administer a blocking antibody to that, and animals regain motor skills, et cetera, et cetera. Again, using the neuropath kit from NanoString, we did see some responses in astrocytes, but again, I want to go back and try the nuke-seq to get the astrocytes out to get a better perspective on what they're doing within the context of these two different models. Oh, and this straight up infection, as a matter of fact, as well.
And potentially neurons, although they may
absolutely
be affected indirectly through
right. Absolutely.
Right. Roberto, you got a question? Roberto?
Yes. Hi, everyone. Yeah, Tom, great talk. I was wondering, you had mentioned that the double transgenic CXCL1 mice had pretty much identical viral control, but you started to see a lot of that pathology and mortality.
Mortality, yeah.
I'm wondering what your thoughts were in terms of what's driving this death after you presumably are getting the viral clearance.
I will tell you exactly what I think is happening, because GFAP not only is expressed in the CNS, it's expressed in the gut. And I would not be surprised if we are getting increased neutrophil accumulation in the gut, in the intestine, and that could be, and I'm just waiting for a student to come in the lab and do this, because we're not experts in gut, but that's what I think the mice are dying from, but I don't know that for sure.
Interesting.
But Tom, the gut's pretty rich in neural tissue. Certainly it's well innervated. And is that GFAP in the neural tissue, or is it in the gut tissue? Have you located it?
I think it's in the neural tissue. I mean, that's why, the gut associated neural tissue. No, we haven't even, that's just from reading papers on GFAP global expression. And so, now that we've got the colony up and running, we can do those experiments now that we're here.
Right. So I'll say we got 60 people. You're a very popular guy, Tom.
Yeah.
Who tuned in. So thank you all for tuning in again. For those who missed the beginning, Rebecca will stand out. Yes, indeed. Very good.
Thank you.
Thanks,
again, thank you. It's great to be back. You have no idea.
Well, I didn't know it before, but great to have you here.
All right.
So,
I will,
yeah, tune in next week for Kai, Cass, and Bron.
If anybody wants any of these CXCL1 double transgenics, don't hesitate to ask me. Okay. Bye-bye.
Thanks,
Tom.
Bye.
Bye.